this is a combination of a lot of different thought processes that have taken place in society. When we were in when we were in New Orleans, we specifically had a strategic meeting on where's the society today and where do we need to be. And one of the things that we felt was we needed to have a more effective way of reaching out not only to our research community but more to the NIH and the regulatory community to give them somewhat of a roadmap of what we think is emerging from the data that's being generated by our colleagues. And so based on that, we sent a flyer out to all the past presidents to help identify what they would consider the top 10 clinical targets that could be validated successfully within the next 5 to 7 years. We got a phenomenal list from them, and that then was taken and sent out to another group of individuals that are actually actively clinically researchers that are doing some of these studies to see if they wanted to add on to that list or reprioritize. That's come back to the society, and we've now taken that and given it to our board of directors to have them put it as an impact, which ones would have the most significant impact. And so they've ranked them independently. Once we complete that, our final objective is to go outside of our society and to the clinical community and get leaders in the field to say what would be the most important thing for you to see happen out of this list. And right now I think we have about 20-something diseases we've identified. And we hope to narrow that down to 10 that we would like to put in a letter and have it signed by all the past presidents of the society and send that to the director of the NIH as we can help guide you based on what's been identified by research actively happening now, data that's continuing to be generated, and in addition, leaders in the field that are watching what's happening, that have expertise in the various levels of gene delivery that's going on, and then give it to him so that he can have a roadmap to help determine how they can best facilitate these going forward. So for example, the hemophilia was identified as one of the top 3 targets. This is, again, there's a lot of excitement in the early phase 1 data that suggests that patients are now out over 2 years no longer taking exogenous protein factor and having expression of the therapeutic gene, indicating that the procedure was successful, the transgene's working over time, and that there may be this benefit attached to it that's very close if we can continue to validate these in the manner that the FDA and the research community would expect. That's one. What's happening here at the NIH today is we're seeing basically the different categories being described: the ocular gene transfer, the neural gene transfer, the cardiac gene transfer, the neuromuscular. So we have these portfolio of families of successes that are taking place that are being discussed today at the NIH. These are probably the ones that are contributing into this target 10 that we again would like to prioritize and present it back to the NIH in a manner of a partnership that we hope to make it successful.